Hey, good afternoon and welcome everyone. My name is Nicole Langer and I serve as the Director of Major Gifts at Northwestern University Feinberg School of Medicine. Thank you all for joining us today for the Northwestern University Medical Alumni Association's virtual webinar series. Today I'm pleased to introduce our speaker, Dr. Lucy Godley, who will be leading a presentation about the Silver Family Blood Cancer Institute, a model for fostering translational research. Dr. Lucy Godley joined Northwestern University in May 2023 when she was recruited to become the inaugural director of the Jeff and Marianne Silver Family Blood Cancer Institute of the Robert H. Lurie Comprehensive Cancer Center of Northwestern University. The Silver Family Blood Cancer Institute was established to strengthen and expand translational research and clinical trials within Lurie Cancer Center. Dr. Godley is a recognized expert in the treatment of patients with blood and bone marrow cancers, especially leukemias and myelodysplastic syndromes, or MDS. Dr. Godley also serves as the clinical director of cancer genetics. This key Lurie Cancer Center program provides inherited genetic risk assessment, surveillance, and management for individuals and families at increased risk for developing cancer due to personal or family history. Please join me in welcoming Dr. Godley.
Thanks so much, Nicole, and welcome everyone. I am going to share my screen. And I'm going to start the show. So hopefully now you can see the first slide. I give you my office number here and my email, just in case at the end we're not able to get to all of the questions. Or if you have maybe a slightly personal question that you want to ask me directly, you're welcome to contact me there. Today, we're going to talk about a really, I think, quite unique and exciting program that we have here at Northwestern that was started by the Silver Family donation. And I really think that this Cancer Institute can be a model for many other groups here at Northwestern and beyond. I'm going to explain what that is in a moment. But first, I always like to start with acknowledgements. And to begin, of course, I have to thank the Silver family. This was a very generous gift that the family gave to Northwestern with the idea of fostering translational research in the blood cancers, blood and bone marrow derived cancers. And in the next slide, the next several slides, I'm going to explain what I hope you will see as a creative way to use this donation to further the work of everyone here. And that's challenging. How do you foster everyone's work at once? I also really want to thank my patients. Over on the left, you see the thank you note that the daughter of one of my patients gave to me, and it's thanking me for caring for her. But really, I thank them, the patients and their families, all of you, patients and families who contribute to research. Because without the patients, we wouldn't have new ideas. And for me, that came during my training when I was taking the personal and family histories of my patients, and I felt like so many of them were programmed to get their bone or blood cancers, bone marrow blood cancers. And they really helped start what has become quite a phenomenon, which Nicole mentioned, the idea that a lot of blood cancers, bone marrow derived cancers, are actually inherited genetic predisposition conditions. So we'll talk just a little bit more about that as the talk gets underway. In my research lab, we have a lot of people who are really dedicated to this topic. And over on the right, you see the current members of the Silver Family Blood Cancer Institute. And I'm going to show you photos of them in a little bit. So as Nicole mentioned, I came to Northwestern in May of 2023 to wear two hats. The first hat you can see at the bottom was to be a hematologist oncologist here. So that's a blood doctor and a cancer doctor, and to be the clinical director of cancer genetics. So our idea here is to really understand why people get cancer, who is at a genetically increased risk for getting cancer, and to provide those individuals, first to find them, identify them, and then to provide them comprehensive cancer risk screening. And that has been very, very successful. We actually have been able to launch a specialized clinic for people with what's called Lynch syndrome. So that's very particular inherited DNA changes that give them risk for um, colon cancers, uterine cancers, and other cancers. And our idea is to set up those types of, I call them disorder-focused clinics for all of the different inherited cancer risk disorders. So we have a team of genetic counselors who are doing an amazing job. And we've started these disorder-focused clinics. That's quite unique, is not usually the way cancer risk is organized. Now you can see the top hat there I call research, and that is where I do my own research. I'm just going to mention some of it today. And I direct what we now call the Silver Family Blood Cancer Institute. And again, that Blood Cancer Institute was charged with fostering translational research, not just mine, but everyone here at Northwestern. And so the Silver Family Blood Cancer Institute does exactly that. We foster the work for all Northwestern investigators who are working on blood or bone marrow cancers. And you can appreciate that as a lot of people. Those are what I call patient-facing investigators, people who see patients and can identify really interesting patients. Sometimes it takes one interesting patient to start a whole new field. We were talking about that a few minutes ago. And then there are other investigators who are what I call laboratory facing. So they never come to the clinic, they never see a patient, but they're working in the laboratory on blood and bone marrow cancers. And it would be very, very helpful to them to have direct access to the patient material. And the Silver Family Blood Cancer Institute fosters all of that work. So how do we do that? First of all, what do I mean by that term translational research? People mean different things when they use that word. And for me, translational research is research that directly touches the patient. Either that the samples come directly from a person, or the results from that work will have direct clinical impact. So this translational research is quite a broad term, and it can mean different things. Now, what I picture here on the left, you see us in clinic, in the hospital, taking care of the patient. Over on the right, you see us in the laboratory. Here, we're looking under the microscope. We might be using different animal models or tissue culture models of disease. And what I put in the middle is my rendition of a chasm or a big separation. These worlds are actually quite far apart. In physical space, they're quite near one another. Northwestern has been able to put its research laboratories in quite close physical proximity to the clinic and the hospital. But in terms of practice and traditions and culture, these are actually quite different worlds. And as I was saying a few minutes ago, the work that goes on in the clinic and the hospital with patient-facing investigators can feel quite separate from the work that's being done in the laboratory. I feel like my job as somebody who stands in both of those worlds and is stewarding the Silver Family Blood Cancer Institute work is really to bridge these two worlds. And the way we do that, I think, is quite unique. We have part of our team, and I'm going to show you the team in a minute, part of the team actually helps write the research studies. And we now have a research study for all the different types of bone marrow derived cancers. So every person with a blood or bone marrow cancer walking in the door has a research study that they can sign and participate in. And we have members of the team who are able to be in the clinic and the hospital. They do the consenting of the patients, and they're right at the bedside when we do a bone marrow biopsy, for example. The bone marrow biopsy is a very precious moment when we're actually putting the needle directly into the factory, the bone marrow, that soft inner part of the bone, and we take the samples. And that's a very, very precious moment when we can get the very, very primitive cells that are in the bone marrow and store them away for future research. We've also been able to foster the laboratory-facing investigators who never see a patient but really want that material. We've really been able to help them write their research studies so that they're able to access the primary patient material. This may seem very, very simple and kind of obvious, but actually the amount of work that it takes to build this infrastructure is really quite a lot. It takes a lot of sophistication to write the research studies in a way that are specific enough that our institutional review board will accept them, but broad enough so that we don't have to bother our patients and their families, every time they come into clinic to re-sign the consent. So the first thing the Blood Cancer Institute did was develop these research protocols, both on the clinic patient facing side and then also on the laboratory facing side, and have those research studies be able to talk to one another so samples can go back and forth, and then actually put into practice talking with the patients, getting the informed consent, getting the samples, and then bringing those samples to the laboratory investigators to allow them to do that work. So these are the people behind the Silver Family Blood Cancer Institute. First, you see Selena there over on the left with the red shirt. She is the leader of the group, and she's really an expert in bioinformatics and analyzing what we call single cell data. She is the person who really helps the laboratory-facing investigators write their studies so that they can access the primary materials that are being collected. Next over, you see Matt. He is now in medical school, so he's not with us anymore. But I think this is very important for you to see that the people who are part of this institute really have goals beyond, beyond for their own career development. And that's really important. We really want people who are very excited and looking beyond at their next steps, and working with the Silver Family Blood Cancer Institute, and say in Matt's case, working directly with patients and helping them sign consent, explaining protocols, collecting samples, was, I think, a really major reason that he was accepted into medical school, because he had direct patient contact and he could talk about what it's like to work with sick people in the clinic and talk to them about the importance of research. Next over, you see Courtney. Courtney is wearing the white lab coat, and that kind of gives you the hint that she's in the laboratory a lot. So Matt and Michelle are off in the clinic more, and Courtney is more based in the laboratory. So when the samples come in to the laboratory, Courtney processes them. Sometimes she makes a permanent cell line out of the sample. That's only if the patients give us, or the research participants give us permission to do that. And then we biobank everything. The biobank is that liquid nitrogen tank that Matt and Courtney are leaning on. And then Michelle is another one of our more patient-facing members of the team. Michelle toggles between the clinic and the inpatient hospital service. And because Matt went off to medical school, we had to replace him. And Catherine over here on the left is our new face. And she's been great. She's been in clinic and really doing a lot of consenting. So the team right now is Selena, Catherine, Courtney, and Michelle. And we're hoping to increase the team by a couple of people in the next couple months. So I wanted to give you some examples of projects that are underway. both patient-facing investigators who are using this team to foster their work, as well as laboratory facing investigators who are able to access these samples. So first we have Dr. Abaza. She works on a process that's very interesting called clonal hematopoiesis. This is the idea that as we go through life, we acquire DNA changes in all of our stem cells, actually, in all of our organs. It's sort of a sign of aging. But we can measure this very easily in the blood system because the blood comes from the bone marrow. So if we've acquired a DNA change in one of our active bone marrow stem cells, those cells will be out into the peripheral blood and we can sequence them. And this process of acquiring DNA changes in our stem cells over time is a risk factor for developing a blood cancer. It's a risk factor for developing solid tumors. It's actually a risk factor for strokes and heart attacks, and for many different inflammatory diseases in the body. And so there's a huge effort in our hematology field in general to understand this process more. And Dr. Abaza is right at the front lines. So she is patient facing. She has a special clinic for people who've been identified as having these DNA changes. And we are able to work with her to get informed consent to save samples. These are very precious samples. We hope to really understand the biology over time. Dr. Altman and Dr. Swarup are my colleagues in the leukemia program. And based on some of the work that we had done on a gene called CHEK2, which I'm going to explain in a minute, they developed a very specific question about how often people with chronic myeloid leukemia, or what we call PH or Philadelphia chromosome positive acute lymphoblastic leukemia, how often these people have inherited cancer risk. Right now, inherited cancer risk is not really on the radar for the doctors for the patients with CML or chronic myeloid leukemia, but based on our work, we think that that's going to change in a major way. So this is an example where our clinic facing investigators know about our more basic research. They got a very specific question, and the Silver Family Blood Cancer Institute staff was able to go out into the clinic and the hospital, find the CML and Ph positive ALL patients, consent them to research. And I think this is going to be a very major insight in our field. Dr. Krismita DeMello actually is in the Department of Neurosurgery. And she's been trying to understand how to make brain tumors more sensitive to immune-based therapy. Many of you may know that one of the major advances in therapy over the past several years has been to harness the immune system. We've always done that in the case of stem cell transplant, but now the number of ways we can manipulate the immune system is really skyrocketing. And brain tumors are relatively resistant to immune-based therapy. But in her work, she identified CHEK2, the protein that we work on, in a totally separate set of experiments. She identified people with problems in CHEK2 actually having rendering sensitivity to immune-based therapy. So this, for me, is a very exciting development. Here you have someone in neurosurgery working on brain tumors whose work is now relevant to us in the blood cancers. And we're working very closely with Dr. DeMello to harness what each of us is understanding in the blood system versus the brain tumor system. Next on the list, we have Drs. Khan and Liu. They work on what's called TP53 mutant myeloid blood cancers. So TP53, some of you may know the name P53. It's kind of famous. It's called the guardian of the genome. It's one of the first proteins that was really recognized to be important in cell death. And some of the most resistant leukemias that we have now have those mutations in the P53. And so Dr. Khan and Dr. Liu are really trying to understand this at a deeper level so that we can develop new therapies that are desperately needed. The next two investigators are a pair of lab-facing and patient-facing investigators who've worked together to develop a pair of protocols for people with chronic myeloproliferative disorders. So Dr. Mello Cardenas is in the Department of Pathology. She's laboratory facing. She's done very, very nice work on the MPNs. And many of you may know Dr. Shamo in clinic sees the patients. And so this is a really nice example where the Silver Institute has been able to work with both Dr. Mello Cardenas and Dr. Shamo, writing the lab facing protocol for Dr. Mello Cardenas, and then helping Dr. Shamo with her patient facing protocol, having those two protocols talk to one another so that we can get samples from Dr. Shamo's patients over into the laboratory where Dr. Mello Cardenas can do some very basic research. Many of you may know Dr. Seema Singhal. She's our patient-facing investigator who works especially on multiple myeloma. And given some earlier work that we had done on inherited risk, we have started a new project here with her on inherited risk for multiple myeloma. Dr. Singhal has been banking samples for many, many years from her patients, and that provides a really amazing patient sample source to ask a very important question that I think is really underappreciated in the field right now. You see Dr. Swarup's name again. He comes up again. In this case, I would say almost more as a laboratory-facing person. So he's like me. He has both an MD and a PhD, and he steps in both worlds. And he has identified, along with Dr. Altman, a new inherited cancer risk disorder, which the more we dig into, the more we think it actually predisposes not just to blood cancers, but also to solid tumors. In fact, we think maybe gliomas, the brain tumors that Dr. DeMello works on. So here's another example where studying something in the blood system may relate to other tumors and maybe even brain tumors. And finally, Dr. Christine Zhang is a laboratory-facing investigator. She also works on clonal hematopoiesis in tight collaboration with Dr. Obaza, and she's particularly interested in one type of clonal hematopoiesis, the most common one, which we call DNMT3A mutant clonal hematopoiesis. And she takes advantage of many of the samples that Dr. Obaza is able to collect. So I just wanted to give you some examples of projects that are very active and underway now that people here, both again, patient-facing and laboratory-facing, see how this is working. We have more and more people asking us for help because it's really quite unique to be able to have very rigorous consenting and have very solid research protocols, and then be able to take those primary samples back into the lab and really understand them. So I just want to give you one example of how our work, which started in the clinic, came into the lab, has now sort of come back into the clinic. And this involves this one particular DNA repair pathway. So you'll all appreciate that DNA repair is very important. If we make mistakes in our DNA, we don't fix them. That's how cancer starts. And as we studied our patients here with blood cancers and looked for inherited cancer risk, we came up with this pathway. We come up with this pathway again and again and again. When you break the DNA, which is a real problem, you know, the DNA is that double helix. If you break both strands, you have a problem because you have a chromosome end and you don't know where it belongs to. This pathway is the pathway that actually compares those DNA fragments to the intact chromosomes. Remember, you have 2 copies of every chromosome, one from your mother, one from your father. So if you break chromosome 9, for example, you still have an intact chromosome 9. And this is the pathway that's supposed to look at the pieces of chromosome 9 and say, hey, you guys go together, and you go together in this order because I see the intact chromosome there. It's a very famous DNA repair pathway called homologous recombination DNA repair. And it starts with this protein ATM recognizing the DNA break, and then ATM talks to CHEK2 CHEK2 talks to P53, BRCA1 and BRCA2, or I usually pronounce it BRCA1 and BRCA2 and others. And these proteins down here, they stop the cell from dividing and say, hey, we have a problem with our DNA. We have broken DNA. We have to fix it. And if this pathway is working well, that happens. But if you have an inherent DNA change in ATM or in CHEK2 or in BRCA1 or 2, this pathway doesn't work very well. And you have a cancer predisposition. As many of you will appreciate, inherited BRCA1, inherited BRCA2 are among the most common that the lay public know about. In Chicago, inherited CHEK2 DNA changes are probably even more common. And as we were doing this work, this CHEK2, as I just said, is one of the most common genes that has an inherited problem in our population. In part, this is because we have so many people with Polish descent in Chicago and from Eastern Europe, but that's not the entire story. Now you can see when we plot all of these different inherited changes that we have from our patients, there's kind of this pile up here of this I157T. And if you talk to a breast cancer doctor, they'll tell you that this particular DNA change is kind of weak for cancer. It's not really considered a very strong breast cancer risk. But if half of our families have this exact change and all of our families have blood and bone marrow problems, then we started to think this particular change, and maybe CHEK2 in general, was very, very important in the bone marrow, maybe more so than other organs. So that was an idea that we had. And we did what's called an odds ratio or burden calculation, where we said, maybe we're only seeing a lot of this because we have so many people of Polish extraction or so many people from Eastern Europe. Are we just seeing this because we're Little Poland? The answer there was no. We're seeing this because this DNA change, the I157T, really has a powerful effect in the bone marrow. This next one, the S428F, that one is common in Ashkenazi Jewish populations. So we said, are we just seeing this because we're a pure Ashkenazi Jewish population? Which of course we're not, but let's argue the statistics away from our favor. And basically here you see a whopping odds ratio of 17. So we are definitely not seeing this change because we're a pure Ashkenazi population. We're seeing it because this change has a powerful effect in the bone marrow. This last change here is the one that's probably most closely associated with breast cancer. And you can see here, we see the least strong effect in the bone marrow. So this says not all CHEK2 changes are created equal. Some have stronger effects in some tissues rather than others. So then we looked in the bone marrow cancers and we said, okay, you have an inherited change in CHEK2. Is there any signal in the leukemia that develops that you actually came from a problem with CHEK2? And remember what CHEK2 does. CHEK2 fixes double-strand DNA breaks. And if you have an inherent problem in CHEK2, you may not align that chromosome, the pieces of chromosome 9 correctly. So for example, if you break chromosome 9 at the same time that you break chromosome 22, this pathway, if it has an inherent problem in CHEK2, it may not put the pieces of 9 together. It may not put the pieces of 22 together. It might put the front of 9 together with the back of 22. And that's what we call the Philadelphia chromosome. So we actually saw a statistical significant increase in what we call translocations or chromosomal rearrangements in the leukemias that develop from the people with the inherited CHEK2. So if somebody has a leukemia with a chromosomal rearrangement, that's kind of a hint that they might have an inherent problem with CHEK2. And there's lots and lots of other data that I don't want to talk about today, but we published this paper very recently, and we kind of said to the world, these inherited inherited problems in CHEK2, they don't just cause breast cancer. They don't just cause colon cancer. They also cause blood cancers, and we need to think about them. And because doctors Altman and Swarup were listening and paying attention to that, they said, hey, wait a minute. What about that reciprocal translocation between chromosome 9 and 22, what we call that Philadelphia chromosome? What you're saying then is if somebody has that Philadelphia chromosome, they might have inherited CHEK2. Well, that is a very famous chromosome, the Philadelphia chromosome. It's found in chronic myeloid leukemia. It's found in the ALL, the acute lymphoblastic leukemias that have the chromosome. And so Doctors Altman and Swarup said to the Silver Family Blood Cancer Institute, can you help us do a study specifically in people with the Philadelphia chromosome? And then we can test whether they have inherited cancer risk and is the number one gene CHEK2. And we're almost finished the study. It's going to have very major impact, I think, in the field, because right now, patients with chronic myeloid leukemia are not routinely being recommended to have inherited cancer risk. And same for Ph positive ALL. So I think once the study gets published, I do think we'll be changing clinical practice. So coming back to this pathway again and again and again with our patients turns out to be it's an extremely important pathway. Over the weekend, I was at an event and I met a family where the mother has the ATM, the father has the BRCA, and their daughter was diagnosed recently, 24 years old, with breast cancer. The daughter got both the ATM from one parent and the BRCA from the other parent. So she got a double whammy of problem in this pathway. And it's probably not surprising that she got breast cancer so young. Now, what about the age? I just mentioned a very young woman with breast cancer. That's very striking to doctors and families, right? Why is my 24-year-old having breast cancer? We've been paying attention to age for a very long time. And here I'm going to focus on myelodysplastic syndrome, or MDS. We've been paying attention to it. Pediatricians have been paying attention to it. And really, all of the MDS doctors have been paying attention to the age. And it turns out at the age at which you present with MDS, when you're diagnosed with MDS, just reads out the inherited pathway, or the pathway in which you have an inherited DNA change. So overall, from our work, about 10% of all MDS is genetic. That's considered very high. And depending on the age at which you present, I can pretty much guess the gene. So if you're a child, 0 to 5 years old, that's extremely young. The average age of MDS, everyone in the United States, is about 72 years old. So why is a 3-year-old getting a 72-year-old's disease? So having a disease like 24 years old with breast cancer way earlier compared to the average certainly makes us think about inherited risk. And certainly, whoops, let's go back. Certainly, seeing MDS in an infant, in a child, makes us think about inherited risk. Now we know they're overwhelmingly likely to have inherited risk in the genes called SAMD9 or D9L. If the person is a teenager, especially if they have only one chromosome 7 in their MDS, they almost certainly have an inherited change in this gene called GATA2. In the adult range and young adult range, the problems are DNA repair, like we just talked about, or the telomeres. The telomeres are the ends of your chromosomes that protect the genes that are sitting on the chromosomes. And then in the average age, 60s, 70s, 80s, we actually have an inherited risk, DDX41. This really kind of blows the minds of the doctors and the scientists, Because the idea has always been, if you have inherited cancer risk, you get disease as a young person. But here you have people in a totally average age who actually have inherited risk. They've lived their entire life perfectly fine. And then in the 60s, 70s, and 80s, they develop their MDS. So this has really challenged the idea of inherited cancer risk. Inherited cancer risk is for everyone. It's across the age, and the age only helps you guess the gene. Well, we don't really play guess the gene. When we do our clinical testing, we test everybody for everything. So we're learning more and more things every day. This idea is absolutely not unique to myelodysplastic syndrome. We've done the same thing for multiple myeloma. If you're diagnosed with multiple myeloma, very, very young. So again, the average age diagnosis multiple myeloma is in the 60s. If you're, decades earlier than that, you often have an inherited change in KDM1A, or that's also known as LSD1. And if you develop myeloma in the average age in the 60s and 70s, we see inherited risk, especially in BRCA2, CHEK2, ATM, also in BRCA1, but more so these three genes. So this paradigm of looking at age to signal biological pathway is something that I think is probably true for all of the blood cancers. And as I explained earlier, the Silver Family Blood Cancer Institute is now working on this project in myeloma to help fill out all of these age ranges across the board like we've done for MDS. So this is sort of the end of my talk. I really think the Silver Family Blood Cancer Institute is a model for translational research that could be replicated well beyond blood and bone marrow cancers. This could be done for all of the solid tumors. It could be done for all of the different disease groups in medicine and beyond, because it provides this essential bridge that bridges that chasm that I showed you before. We have rigorous consenting. We only do things that for which we have permission, like say making permanent cell lines. We ask everybody. Some people are okay with it, some are not, and we respect everyone's decision. So everybody walking in the door, we hope in the future, well, certainly now for blood and bone marrow cancers, but we hope in the future all individuals who come will consider participating in research to help us understand their diseases more. And we do biobanking right now, but by that I mean we take primary tissues, put them in the freezer, and store them for when someone has an important research project. We look at inherited cancer risk, as I've indicated. We look at acquired DNA changes, as I've indicated. We call that now clonal hematopoiesis. We are also really interested in the microbiome. Those are all the bacteria and fungi that live on your body and in your body and really mediate your risk for disease. And then, of course, the environment and the social pressures that we have in our lives, that affects us from a biology standpoint as well. This is something Dr. Khan is very interested in, and she's a leader in this field. And these are all layered effects, all from the DNA you're inherited to what you acquire over your lifetime, to then these other influences that are more from your environment. And once we understand all of these factors, I really think we'll be able to have very, very effective therapies. And our goal is to design new clinical trials. My ultimate goal is to put us out of business. If we really understood who was at risk for cancer and we understood all the different pathways and mechanisms by which someone got cancer, we should be able to interrupt it. And so that's really, I think, the ultimate goal for the oncologists is to put ourselves out of business. And with that, I just want to end again by giving you my phone number and email address and thank the Silver family again for their generosity and look forward to your questions. So thank you all. I'm going to stop sharing here and we can open it up to questions.
Thank you so much, Dr. Godley. I will help kick us off with the Q&A portion of the webinar. We received one question asking about MPNs. So can you share any focus at the Silver Family Blood Cancer Institute about MPNs?
Right. Well, I tried to do that when I showed you that nice pairing between Joanna and Jamil. Dr. Mello Cardenas is the laboratory-facing investigator in pathology. Dr. Shamo is the patient-facing investigator in hematology. And this is such a beautiful example where when we started working with both of them, there were no studies. There was nothing a patient could sign. But Dr. Shamo is very, very smart. She has a lot of really interesting ideas, and she really wanted to get that off the ground. And Dr. Mello Cardenas is doing really beautiful basic biology, but she needs to get her hands on the samples. And so Selena from the Institute was able to work with both of them and then essentially put a link between those two protocols so that now patients with myeloproliferative disorders are able to participate in research. And I don't want to speak for Dr. Mello Cardenas and talk about what she's doing, but I think she's really very interested in the microenvironment and how the bone marrow garden, if you will, fosters the growth of these cells. The ultimate idea would be to interrupt that process. And Dr. Shamo would be able to translate Dr. Mello Cardenas's basic findings into a clinical trial. That's where we're trying to get to. So we've put all the basic building blocks in place to allow that.
Thank you. Another person asked, how long is a bone marrow sample kept?
Yeah, that's a great question. Clinically, when we take a bone marrow sample, when people are having bone marrow biopsies, and we only do bone marrow biopsies for clinical purposes, there's a specific question that's being asked from that bone marrow biopsy. What is someone's diagnosis? Are they in remission, et cetera? And those cells are not propagated for a long time in the laboratory. So the most they're probably alive is about 2 days in the laboratory. The samples are processed very quickly in pathology so that the clinical testing can be done. If someone has given us consent to bone marrow samples, we generally process them and just freeze them. We freeze them in that liquid nitrogen tank, which I showed you, and that's considered the most stable way to freeze living cells. We do freeze them in a living cell capacity. It's harder to do that because we have to put sort of stabilizers in the sample so ice crystals don't form and kill the cells. But that allows the cells essentially to be there forever. Now, if someone decides they don't want that to happen, they can withdraw consent, which we hope people don't do. But if people do decide they don't want to participate anymore in the research, then we go in and we pull all the vials from the liquid nitrogen tank and destroy the samples. But we've used samples that have been in storage more than 20 years, for example, to do some of our work. And this is incredibly important to be able to go back to samples over time. So when treated properly and stored properly, cells can live forever. Now, once you thaw the sample, it won't live forever. So that primary sample will die within a few days. But the cells will be alive again, if you will, once you thaw them. We can make permanent cell lines, as I've indicated. We only do that with patient consent. Once we make a cell line, that cell line could live forever if it were propagated in the lab. So hopefully that answers the person's question.
Great. Another person has asked about thrombocytosis, which they understand to be a form of blood cancer. Can you share anything about studies being done at the Silver Institute that are pertinent to heredity, age of acquisition, anything like that?
Yeah. So the word thrombocytosis actually only means an elevated platelet count. And the most common reason that someone has thrombocytosis is iron deficiency. So this is very, very important. Having an elevated platelet count is not equal to cancer. And the reason that iron deficiency causes an elevated platelet count is kind of interesting. When you're iron deficient, you're anemic. And when you're anemic, the kidney senses that you're anemic. Why the kidney? It just has to do with evolution. It's kind of interesting. And the kidney senses the anemia, and the kidney sends a hormone over to the bone marrow called erythropoietin. It was discovered at the University of Chicago. And that hormone is like, hey guys, we're anemic, make some red cells. And the cells that form the platelets have the receptor on them for that erythropoietin. So when you're anemic and the kidney's making high levels of erythropoietin, it stimulates platelet production. So the most common reason, again, for an elevated platelet count is iron deficiency. So you definitely have to make sure that someone's not iron deficient. But the person is correct. A lot of times for our patients, it can be a sign of a bone marrow cancer. Often those myeloproliferative disorders that we were talking about before, but sometimes even a particular chromosomal rearrangement, like we were discussing these translocations. There's a particular, we call it an inversion of chromosome 3, where one piece is just flipped around backwards, and that elevates the platelet count. So there are lots of reasons why someone can have an elevated platelet count. A few weeks ago, I had a mother-daughter pair with an elevated platelet count, and the local hematologist said, hey, this is kind of weird, a mother-daughter pair with elevated platelet count, what's going on? We actually identified them with an inherited DNA change that's quite rare. We actually just published, even today, it came out officially, a series of people with an inherited change in this gene called JAK2. Now, a lot of you on the call know JAK2 because it's inherited in the myeloproliferative disorders in an acquired way, meaning you're born fine, but somewhere along the way in your lifetime, you get a change in the JAK2 gene. This V617F is the classic one that's acquired, and it causes myeloproliferative disorders. It causes elevated platelets, for example. But we've identified a few patients within our system who have a different change in JAK2 that's actually inherited. And we just published that case series, and that case series, along with others that have been published, are, again, going to put this idea on the map when doctors see a JAK2 change that isn't the common one. Then they're going to maybe start to think about inherited risk. So there are lots of different reasons you can have an elevated platelet count. Some of them inherited, some of them just related to, say, iron deficiency, some of them acquired. So that's what we call the differential diagnosis. That's what makes patients interesting, because you're like puzzles for us. You come with an elevated platelet count, and then we have to figure out what's going on.
We had several questions about AML. So how much is being done about AML? What are the expected causes of AML, and what component is genetic?
Yeah, great questions. A huge amount of research in hematology is being done for acute myeloid leukemia. How much is genetic? We have not actually done a study of acute myeloid leukemia. I showed you the data that we had from myelodysplastic syndrome. We chose myelodysplastic syndrome for maybe a somewhat random reason. So I can answer the question for MDS. I cannot answer it for all AML. But it is going to end up being a pretty large percentage, probably anywhere from 10 to 30% of AML is genetic. And that's a big range. But the blood cancers are very, very genetically determined. If that AML has one of these chromosome rearrangements, that may make it more likely that there's an inherited predisposition. We need to do the studies. One answer can be people are born with a risk. They may not know it when they're born, that they have a risk for AML. And then definitely we know that as you go through life, if you acquire a DNA change in particular genes in your bone marrow stem cell, that's what we call that clonal hematopoiesis, that is definitely a risk factor for developing acute myeloid leukemia. And now the big area of focus, I would say, is, okay, we know who's at risk. We know who has inherited risk. We know who has the clonal hematopoiesis. How do we slow down the process? We want to prevent AML. And so there's an enormous amount of research going into that. And I do think in my professional lifetime, we will see preventive strategies for people at elevated risk, either because they were born with a risk or they acquired that risk in their lifetime.
Someone was curious to know what proportion of seniors with MDS have the DDX41 gene.
Yeah, that's a great question. If you take all acute myeloid leukemia, so all ages, about 3% of all leukemia is derived from inherited DDX41. 3 out of 100. That may not sound like a lot, but it is really quite a lot. That one gene is responsible for such a, a detectable fraction, so to speak. DDX41 was only discovered in about 2016 by looking at the families. By finding families with multiple cases of leukemia. Many doctors have told these families that leukemia is not genetic. I hope that people aren't saying that anymore. And finally, in 2016, we had enough of these families that we could identify the connection between DDX41 and the leukemia risk. DDX41, along with CHEK2, these changes are extremely common in populations. So both DDX41 and CHEK2 at the population level are, again, on the order of 3 to 5% of populations. That's really, really common. And what I say to people is cancer is a common disease. A lot of people get cancer. And so this idea that we've had in our science world that these DNA inherited changes have to be rare doesn't fit with cancer being common. And actually, again, for DDX41 and CHEK2, these are common changes that are, especially in certain populations. Now, again, the trick is, now that we know who these families are, how do we prevent it? We don't just want to watch it happen. We want to develop strategies to prevent the cancer. And that is definitely where our thinking is going. We don't have anything to give people right now, but everyone is so generous with their materials. And I really, really have great hope that we are going to be able to understand how you go from the inherited DNA change to the leukemia and then intercept it and prevent it.
Do you take samples of patients outside of the Northwestern Medicine hospital system?
I'll say a little bit yes and no. So it depends on what someone means by the sample. But for our inherited cancer risk, we can consent anybody in the world. They don't have to have a medical record number at Northwestern, so to speak. And we do collect families all across the world. Whenever somebody signs consent for our research, it's really helpful if we have a sample from them, because without a sample, we can't do too much. And so we collect a variety of different samples from people outside of Northwestern. In general, though, our Northwestern patients, if they sign a research consent, those samples are collected here and stay here. So that liquid nitrogen tank that I showed you, those samples stay here. We don't ship those samples anywhere. Right now, we are not sharing the data with anyone outside of Northwestern, so it's kind of within the family, so to speak. Not all of the research protocols that I described to you allow consenting outside of the institution. So that question, it kind of depends on exactly what's the research study that would be appropriate for that person.
Someone wanted to support you or the research that you're doing. What are some ways they could go about doing that?
Well, the first thing is participate in the research. So, and generally, almost everyone we approach in clinic says yes, even if not initially. Sometimes there's a lot going on in clinic. People are sick. They're worried about their health or their family's health. And sometimes when we ask about research, it can feel kind of like a distraction. Like that's not why I came here, and that we completely understand that. But a lot of times over time, as they realize this is a teaching hospital, it's a research hospital, um, they understand the importance. They understand the importance of prior people participating in research and how that has established the current standard by which they're being treated. And then they feel, hey, somebody did this, so I could be here getting this care. Let me do what I can to contribute to world knowledge. And so people have been incredibly generous, as I've tried to indicate today, with their samples. So that's number one. Number two, we need patient advocates. So I just, for example, wrote what's called a letter of intent. It's like a pre-application for a very large grant that would bridge the United States with the United Kingdom. And in the requirements for that grant, we had to name patient advocates. And so having people who are willing to be part of research at that level, designing the research studies, participating with the scientists is incredibly important and helpful. And then finally, obviously, finances. We don't want to talk too much about that today because that's a whole big topic. But I think many of you appreciate the constraints that are put on science right now, especially. And philanthropic donations are often what allow new ideas to start. Certainly for myself, the inherited predisposition work, the inherited cancer risk work, when I started proposing that this was a thing in the blood cancers, people laughed, like literally laughed. And if it hadn't been for philanthropic study money and to support those initial ideas, there's no way it would be where it is today. So sometimes the philanthropic donations allow the new, I think exciting, but one could also say maybe more risky ideas. Certainly, you can see the Silver Family donation, what that has allowed, how that has multiplied out to affect so many people here. Sometimes people want to donate their funds to a particular project because that disease is very important to their family. But sometimes, like the Silver family said, take this and multiply it out to all the investigators. So there are a variety of ways that people can participate and help.
Okay, I think we have time for maybe one last question. Someone asked about, if a parent in the family has CLL, are the children then at a higher risk of also having CLL?
Yeah, well, let's expand the question not just to CLL, but let's take any blood cancer. The answer is yes. CLL in particular is considered to be one of the most heritable of the blood cancers. And yet, when those families have been studied, at least in the past, the obvious DNA changes um, that confer that risk have not been identified, but that is changing. And so, for example, we have the gene POT1, POT1. It's one of those telomere genes um, that is now recognized as being an important inherited risk for CLL. We've also seen CHEK2. When we analyze the database from the United Kingdom, we can see a statistical association between inherited CHEK2 changes and chronic lymphocytic leukemia or CLL. So I think we've always known from family studies that leukemias are heritable, and now we have actual genes that we can look at and study.
Great. Well, I think that is all the time we have today for questions. So I want to thank you again, Dr. Godley, for your wonderful presentation, and hopefully we can do this again sometime soon. And thanks
Well,
everyone on the
yeah,
web for
thanks
joining
everyone.
us.
Thanks for having me. I hope we can talk soon.
Thank you.